All right, friends, this is the third lab. Again, not the third one we did. We did this as part of our mitosis and DNA replication unit. And indeed, many people forgot to do this because I handed it to you and said, do this at home online. And of course, that's the death sentence for any lab. So let's talk about it now. Essentially, in this lab, uh, the honestly, we didn't really do a meiosis portion, though the lab, we actually did the meiosis portion of this in class together. This was just a modeling activity. I don't know if you remember, we modeled the chromosomes going through mitosis and meiosis using markers and whatnot, and that's fine. But the real part of this activity that's an actual lab that you did was you went online and looked at photos, very tiny little photos in a program that were in different stages of mitosis, and you counted them. If we had done this lab for real in class, I would have given you a slide of an onion root tip, and this is not insignificant. If we want to see mitosis, we have to look where it's happening. There are some cells where a lot of mitosis is, or some regions of living things where there's lots of mitosis going on, and other regions where there is not. Places where there's a lot of mitosis going on are in the growing tips or meristem, here's a plant term, meristem tissue of plants, the tips of roots, and the tips of shoots of plants is where they grow. They don't grow in the middle in the tree trunk, they grow at the tips. So if we want to see lots of mitosis, a good place to look is in a tip of a root or a stem. And the onion is an easy organism to look at, and so they are commonly used. What you would have done if we had done this in class is you would have looked in a microscope at the tip of the onion root in the meristem tissue and seen lots and lots of cells in different stages of their cell cycle. And you would have counted how many you were seeing. And you would have done this for like 3 fields of view in the microscope, and you would have done hundreds of cells. It's quite tedious. So I gave you the online versions because it's easier. You look at 36 cells, but you get an idea of what you need to see. Now, the idea is that if you look at a root tip under the microscope, and there's the little root tip area you'd be looking in, you would see cells that were dead. They have been preserved. So they aren't actively dividing, but if you count enough of them, you can say, okay, at this snapshot, there were 100 cells in interphase. There were 7 cells in metaphase, and you can get a sense of how much of a cell's life on average is spent in each stage. The more cells in a given stage, the more time the cells must be spending in that stage. The fewer cells in a given stage, the less time. And the object of this lab was to see if you could determine how much time a cell on average spent in each of the stages of mitosis, plus interphase. Please remember that interphase is not a stage of mitosis. Mitosis is cell division. Interphase is when the cell is not dividing. Okay? Now, all of that said, okay, we already talked about that. Count the number of cells in each stage. Okay. We did not do this crossing over lab, so I'm going to ignore that because it's not asked about very often. Just that occurs in meiosis only, and we're just going to skip over that. Now, please know, unfortunately, the way this is laid out, this suggests that interphase should come under mitosis. It should not. It is its own thing. It is not part of mitosis. Mitosis is these 4 stages that act to separate those chromosomes from each other. Interphase is when the cell is working and doing its job, or it's replicating its genetic material. Okay. Meiosis, on the other hand, is to create sex cells. We go through two divisions. Okay. What you should have concluded from this lab, in addition to understanding about mitosis and so on, is that the cell spends its vast majority of its time in interphase and much less time in all of the other the stages of mitosis. It spends a little bit more time in prophase and then very little in meta, ana, or telophase. And I don't want to take the time to get into the mitosis meiosis comparison here. Let's look at, this is asking you about meiosis. I'm not going to do that. This is all we're going to do with this. I do want you to remember that mitosis is talking about cloning of cells, making exact duplicates. There is a question associated with this lab that asks, why is mitosis better referred to as nuclear replication than it is cell division, even though we always call it cell division? And that's because we are actually copying the genetic material and then distributing it to two daughter cells. And of course, remember please that meiosis is cutting the genetic material in half to create sex cells, so that's a very different process. I will take this moment to talk to you about the cell cycle, just real quick. Please do remember that, and it's how it's controlled by cyclin and cyclin-dependent kinases and all that, CDKs. But if this is your cell cycle, please remember G1, S, and G2. G1 would be where the cell is growing in size and where it is preparing to make copies of the DNA. Synthesis is where we are going to copy the DNA. We're going to go from being a single-stranded to being a double-stranded chromosome made of two chromatids. Remember that. And then G2 is where we're going to be preparing for the actual stages of mitosis, of cell division. We'll be creating microtubules, the spindle fibers, and all of that. Do remember the purpose of the spindle fibers made out of microtubules that latch onto the kinetochores at the centromeres and pull those chromatids toward the poles of the cell, and so on. So do remember your stages of mitosis. Remember what's happening in them, lining up, which is being shown here. The chromosomes are lined up along the center line of the cell, the equator. The anaphase is where the spindle fibers are pulling these chromosomes to opposite poles of the cell. And telophase, we're going to remake our nuclei again. The prophase of mitosis, remember the chromosomes are just getting thicker before they as they go from being chromatin to being the shorter, thicker chromosomes that we're familiar with. Remember the difference between plant cells and animal cells. Animal cells, we see what is called a cleavage furrow during the cytokinesis phase, which comes right after telophase. So right after telophase, we'd have cytokinesis, where we see the pinching in due to the actin microfilaments. The actin microfilaments will pinch in almost like somebody's tightening a belt, and we split the cell's membrane and cytoplasm in half, whereas a plant cell is going to be different. A new cell plate will simply form between the two halves of the plant cell because, of course, a cell wall cannot pinch in. All right, that's kind of a helter-skelter review of mitosis and meiosis. Again, if anything is unclear about that, please let me know. But otherwise, that's your AP Bio Lab 3. Thanks for watching.